לא יודע אם הוא ימשיך מאותו מקום.
נכון. כן, אז אני פשוט צריכה להחליף את T ב-reset במקום זה.
לא. FTP שלה.
עכשיו, הדבר היחיד שאני נראה לי יכול להתעלם ממנו, אבל שבגלל שזה נורא קטנט, לדעתי זה היה עד כדי 2πk, משהו כזה, לא? יכול להיות אני טועה, אבל נראה לי שאפשר להתעלם ממנו.
רגע, הספק משתנה FTP כמו שאמרנו, ואז נתקע בבעיה של T. אחרי משתנה FTP כמו שאמרנו, וזה הגיע כאילו בדיוק לביטוי הזה. מה שקורה זה שאת מכתיבה את ה-T כאילו במקום שלו במשוואה שתקבל, CVT שווה משהו, כאילו הראש של טנגנט של משהו כזה, ואז צריך להגיד את ה-T שהוא בעצמו טנגנט כאילו של משהו, ומקבלת משהו שאת אומרת, אני לא יודעת מתמטיקה, זה בטח לא נכון. אבל קומוניקציה, לא יודעת את התרגיל, אבל נראה לי
שעדי אמר שאפשר להגיד שהשבר בסוף. זה לא יקרה. לא, אל תגידי ששבר בסוף. כן. מה שתדעי, את יכולה להשאיר את השטחים שאת מדברת עליהם רק אם יש כמו שכתבת, אבל אם זה נכון מספיק או לא.
בעייתי לדבר מסקנות שאני מסיקה בסוף.
לא, זה תגידי.
לא.
אוקיי. רגע, זה הרמקול, זה נכון? בזה אני משתמש בדרך כלל או משהו אחר? אוקיי. טוב, אז זה השיעור האחרון שאני נותן לכם לפני יום רביעי שבו אתם נותנים לי, או לכם, את השיעור. אז דיברנו בפעם שעברה על דרך לייצג תאי עצב מבחינת המודל מתמטי שלהם, על ידי זה שאנחנו ממדרים, נקרא לזה ככה, או מחלקים את העץ הדנדריטי. סליחה? Sorry. So last time we spoke about the way to represent neurons, dendrites and axons by what we call compartmental modeling, which allows me as an anatomist first to reconstruct a piece of dendrite, let's say, then decide on the sampling points, because of course I cannot sample every point, so I discreticize the neuron into pieces, one and another. So delta x is my distance piece of sampling, and then I have to have at each location my 3D XYZ coordinates plus diameter, and eventually I can get a cable representation, a continuous representation, assuming that this little piece has this length and this diameter, and this other little piece has this length and this diameter. And in principle, one can solve the cable equation for a continuous case like this using some systematic way to iteratively solve the voltage at any given location for any given input But today, we usually do not solve it analytically, but rather solve it numerically, meaning that each of these cylinders, each of these cables, is represented by an isopotential piece of membrane. So you assume that all this piece of membrane is short enough to be called isopotential, to be assumed to be isopotential. So this is an RC isopotential representation of this short enough, electrotonically short enough piece of a cable with a given diameter and length. So this will be the membrane area here, and there will be axial resistance, there will be resistivity between this location and the next location. So this is the compartmental representation of the continuous case. So this is the continuous case, and this is the discretized case. Compartmental modeling, which is very popular today. You can discuss it at the level of single cell. So each compartment will represent a piece of dendrite or even a piece of spine, as I showed you last time. So you can say, this is my resolution of a compartment. This is the membrane, head membrane, the membrane of the head. This is the axial resistance of the neck. So I can, depending on my resolution of delta X, I can represent a very complex dendritic tree, depending on my question. But I can also decide, as many neural network people do, I can also decide to call all this a compartment, so the whole neuron is one single compartment, the whole neuron. And then I have a compartmental modeling, like, for example, Chaim Sopolinsky is doing when he's trying to solve dynamical systems, many neurons connected to each other. So each neuron can be represented as a single compartment. All the synapses are collapsed into this one single compartment. And then you solve maybe 1,000 compartments or 10,000 compartments, depending how many neurons you have in your system. Not at the resolution of a compartment of a dendrite, but the compartment becomes the whole neuron. So the resolution of compartmental modeling, depending on your question of interest, yes? Who is your compartment, so to speak? Okay, but this was a methodological issue, just for you to know that today I can take any complex dendritic tree and ask particular questions, either local or more global, and I can use the same compartmental numerical method to represent a whole network of neurons, each neuron by a single compartment, linear or nonlinear, spiking or non-spiking, depending on the type of neuron you want to deal with. Okay, so this was basically the cable theory, which was based on the passive assumption. And as you know, we have a lot of nonlinearities. I mentioned it already before. So just to mention something that you all know, just because the fact that we could, in the last 20 years, use this Dick video microscopy, differential interface contrast video microscopy, I can see cells, I can see neurons, individual neurons in a slice. And not only the neurons, but I can see also processes like dendrites and axons sometimes. And so I can patch it in a very systematic way. And the real breakthrough came about 15 years ago. This was 1995, so it's more than 15, or 1994, where Stewart and Sackman succeeded for the first time systematically, visually guided, to be able to patch both the dendrite here and the axon in a slice of cortical network, layer 5 pyramidal cell. You can see a little bit of the basal dendrites here. You can see maybe a little bit of the axon coming out. In this case, they stained the cell. So they injected a dye into the cell through an electrode. So this is the somatic electrode. This is the dendritic electrode. And so they did something nice just to make sure that your dendritic electrode and somatic electrode are in the same neuron, because you don't really know. When you patch a dendrite here, you patch a soma there, you're not sure 100% by looking, unless you really follow the process. So this was one way to inject a green dye here and blue dye here, and to see that the dye mix with each other. So that means that it's the same cell. Okay, so this was a very beautiful work. Really impressed me a lot. And actually, it was the first time to demonstrate this, what we call the backpropagating action potential, which I hope you heard a lot about, meaning that if you patch the dendrite here and you patch the soma here, and sometimes you even can do, some people can do it today, which is in some sense absolutely unusual expertise to be able to patch in the axon, which is very thin, in the initial segment of the axon, in the dendrite, in the soma together, and you stimulate, let's say here, so you mimic a synaptic input to dendrite, let's say. So you can see that you stimulate the dendrite, and the dendrite become depolarized first. Yes, you stimulate the dendrite. The dendrite become first depolarized. This depolarization attenuates to the soma, as you know from cable equations. So this is essentially almost passive from here to here. But suddenly, because you have a somatic electrode, you see that the action potential was initiated at the soma, although my stimulation was at the dendrite. It was initiated at the soma, and then at the dendrite you see this phenomena for the first time in a systematic way because you have an electrode here. But even further, if you would have had an electrode at the axon, you can see that it is actually the axon that fires first, and then the soma, and then the dendrite. So the order of things in time is that basically independent where you stimulate. You may stimulate synaptic input coming here, synaptic input coming here. Basically the hottest spot is the initial segment. You can see it here. So it takes over and fires first. The axon fires first. And then the action potential backpropagates from the axon to the soma, from the soma to the dendrites. But we were used for many, many years to record only from the soma. And so we thought, so to speak, not really thought, we already knew that the axon is hot from other studies, but this is a direct demonstration that the axon is the first. It's also higher action potential, a larger action potential, and so on. So we know today that there are not only more channels or more dense channels here, but also a little bit different sodium channels with very fast kinetics and so on. I'm not going to go into axons, but axons are gaining more and more focus these days because it is clear that it is not just a simple yes or no spikes, but you can modulate the spikes in many ways because there are these interesting sodium channels in the axon. I'm not going to talk about that, but just to complete the view that neurons are very interesting electrical devices, as you can see. It's a distributed electrical device, so different things can happen in the same microchip at the same time, so to speak. It starts here, backpropagates there, even though the input may come here. But the interesting thing in this context that I wanted to discuss is this backpropagating action potential, which is the first time showing that the action potential is really not only going forward, as we knew for many years from Cajal. Remember these arrows that I showed you. Cajal thought the direction of information flow is this direction. Now I'm showing you that there is also backpropagation of information from the soma, axon, soma, dendrite. And the question of Stewart and Sackman was whether this signal is passive or active. Because you know that you have a big signal here, like a spike, it would propagate backward anyway in a passive cable. The question is it passive or active? And so what they did is something really very beautiful. This is another way to show it. What they did is really very beautiful, which I'm going to show you in a second, but this is another demonstration from another group, just being able to record at several locations, which is really, really very difficult. I can tell you that this experiment, I don't know if there are more than 2 people in the world that can do this today. It's really, really difficult to impale at several points, but then you can see the order of time, the green first, the blue second, and then the dendrite third. Okay, so what they did is very beautiful, you know, because you want to ask yourself whether this backpropagation is really passive or active. And in order to do that, active meaning that maybe this signal that you recorded at the dendrite is enhanced, is boosted, is amplified on the way backwards. So are there active channels that help this backpropagation to become this size, or maybe it's just a passive phenomena? So in order to do that, you have somehow to make the dendrite passive. Yes? You just don't see, but the red one would have been smaller than that. So it attenuates backward. So the backpropagation action potential, as you can see, is not a full-blown action potential. It is not full active action potential, but the question is whether it's semi-active, weakly active, or whether it's completely passive.
Why don't you see it directly, because the blue line is lower than the orange, so something non-linearly should happen.
How do you say it again? What did you say? The blue one is the somatic action potential.
Below the arrow.
Yes.
So it should be very low, but actually it's very high. So something non-linear happened.
I see what you say. Are you saying, if I understand you, are you saying that if it would have been completely passive, if it would have been completely passive, I would see, let's say, this action potential in the soma, and if it would have been completely passive, you say, I should see something like this. It cannot grow above that. That's what you're saying? That the fact that my backpropagating action potential is above the signal here, you mean it tells you that it's already active in some sense. Is that what you're saying? Okay. Okay, so you're right. There is a signature here of maybe some nonlinearity. Note, by the way, and that's something that if we talk about cable equation, this is nonlinear cables now. We are talking about nonlinear cables now. At least the soma is nonlinear cable because it generates action potential, so it's not passive. But you know, the fact that you have something that pushes you, that hyperpolarizes you fast here, just because of the active potassium channels at the soma, even in the passive case, you can get an action potential that goes down. And even if it would have been if this would have been a hot soma and a passive cable, completely passive cable, and at the soma you will get this action potential, here, if it would have been completely passive, you still could get something that looks like that, above the action potential. Okay, because this part is an active, pushing down actively the voltage towards the potassium battery, and if this would have been completely passive, this could happen. In a complete passive system, you're right, that if I give something here, everything else should be below. So as you go away, it will look like that, as we showed last time, with roll, but so in a completely passive case, if you start this at x equals 0, let's say here, you cannot get the current source is here. This is the current source. You inject the current here, you cannot get the voltage at other location above, because this would mean that the current source suppose it was like this means that the current flows this direction, because it's higher here and lower here. So this cannot happen in passive case. So you're right in principle, but because the action potential is doing this, even in the passive case, it could be, even if the cable would have been passive. So directly, you don't really know completely for sure whether this is passive or not. You really have to prove it directly. And that's what they did in a nice way. What they did is really they put into the dish where the network is, they put TTX, hoping that they will block all voltage-dependent sodium channels with TTX. So this becomes a passive, not only TTX, they put other drugs, hoping that they block all active channels, and so they make this dendrite a passive cylinder. Now, and that's the beauty of the thought, they injected voltage clamp in the shape of the blue spike. So the voltage clamp, the soma, now hopefully everything is passive. Okay, it became passive. So I now mimic the spike. By injecting voltage in the shape of the spike that was there when it was active. Is it clear? So this is voltage clamp. It's a transient voltage clamp, yes? It's a voltage clamp that follows if you have a fast enough voltage-based system. You can really model, so to speak, by the voltage clamp the spike, and then you can see now that everything is passive, how much of this simulated spike or mimicked spike is seen at this location. And you can see that much less of what is really actually seen is seen when it's completely passive. It would have been something like that, let's say. Okay, so the difference between this, which is the passive backpropagation, and this one is the boosting, the amplification, due to the fact that, and that's what they showed later on, that in the dendrites of layer 5 pyramidal cell, you have active sodium channels, voltage-dependent sodium channels, in the dendrites, low density, very low density, but still enough to be able to boost, to amplify the backpropagation, rather than having it passive, having it boosted above passive. Is it clear?
But they don't have the inactivation process?
Why? Yeah, they do. It's a sodium channel.
Then it also have the inactivation process, pushing down.
You mean here? You mean?
Or this is only a decay, a passive decay.
Are you talking about the tail? Are you talking about this? Okay. So again, let me make it clear. You know that in the Hodgkin-Huxley model, the classical Hodgkin-Huxley model, the action potential grows because of opening of sodium channels, and it closes because of two things. The hyperpolarization or the repolarization, as it is called, is due to both the inactivation of sodium but also the activation of potassium. And this is what carries the spike below the resting potential. Okay, so if, for example, if I would block the inactivation, if I would block the H function, still I will have the spike ending because of the potassium. Okay? Here, you have mostly sodium. So the sodium opens and the sodium closes, but it doesn't carry it below resting because the potassium is not powerful. Okay, but it is with regular sodium. So apparently what they found is the same sodium channels that exist here generated in the cell body. Remember that most of the channels are the industry of channels, the industry of proteins is being done at the cell body and being sent everywhere. Apparently they are being sent more here, which is an interesting thing. How do you know I'm a channel now? A sodium channel. How do I know that I should go here a lot and not here less, or less here than here? So what is the directionality of channel distribution? It's interesting, yes, because it's being produced here. So there is somebody who tells me don't go so much there, but still go. So there is a little bit of here, a lot of there, less here. So there is a non-uniform distribution of ion channel density of a particular channel type, which is a complete still puzzle. Who is the one who is really managing not only the type of channel that is being produced by the genes, but where these channels are being produced, reside? Where do they go? So as I said, a lot of the sodium channels are going to the initial segment. A lot of the sodium channels are going to the node of Ranvier, between the internodes, into the node of Ranvier. So there is a very particular design of channel densities in axons, but also in dendrites. So that's what I wanted to say. Yes, the dendrites are also hot, semi-hot, weakly hot, pocher, medium hot, the dendrite. And this is why it is not carrying full-blown action potential, because if it would have been like an axon, in terms of hotness, it would be the same spike everywhere. But you don't want the action, apparently, you don't want whoever designed it, evolution, don't want the dendrite to be hot, very hot. Otherwise, because if it would have been hot, it would flush, the spike would flush the dendrite and will reset all the inputs while it is there. So you will have a period of time with flushing the input, killing everything that is there. So you don't want the dendrite to be an axon. But still you want the dendrite to be, apparently, somewhat hot, weakly hot, so that the backpropagation action potential will travel backward actively, semi-actively, in order to to notify the synapses something that maybe they succeeded to generate an action potential. And this is all this issue of plasticity, yes? You know that the backpropagation action potential is absolutely critical and very, very important signal for handshaking between the soma or the axon output and the input that originated the action potential. So this is a backpropagating, very important plastic signal, telling the synapses, the excitatory synapses, that they succeeded or not to generate an action potential. So I don't know if you heard about spike timing dependent plasticity, STDP, and the STDP depends on this backpropagation action potential, which is semi-active. Still, there is an issue, because suppose you are a synapse here, very distally, this is layer 5. I'm talking specifically about layer 5. Not every neuron has this backpropagation action potential. It's not a general phenomenon. But in layer 5 pyramidal cells, in rat, in mouse, and also in layer 2, 3 pyramidal cells, we see this backpropagation action potential, but this action potential does not reach very distally because it attenuates. So you asked before what would have been in the red electrode. It would have been smaller. What would have been in the, if I had an electrode there, it would be very small. So who is telling the synapses that they did something good? Who is talking to these synapses? Is it clear? Who is telling the synapses that they succeeded to act, that there is a correlation between their activity and the spike output? Who is telling them?
Wait, but does that mean that you can't have spike time independent plasticity for those synapses that are
If this would have been the only mechanism, you cannot do that. There is no, because nobody tells them that they did or not. There is no correlation between the activity. If this is the signal, I just wanted to tell you that there is another signal. But if this would have been the only signal, then you would not be able to know as a synapse whether you did or you did not succeed to generate an output. So that's a very important issue, you see, because this was an open issue. How do the synapses here, in a correlative way, in a predictive way, because you have to predict, that's what Tali Fishby would like to tell you, yes? You have to predict that I'm going to succeed in making a spike, and somebody closes the loop and tells me that my prediction was correct or not. And then I change accordingly. So this backpropagation action potential is a signal that ensures, that's why I call it handshaking, because I shake my hand. So I'm sending a signal, I'm a synapse. I'm sending a signal and somebody tells me what was the outcome of my signal. Was it a spike or was it not a spike? So this is the spike time independent plasticity.
So it has also an implication to the propagation from the dendrite to the soma, because if the dendrites are also semi-active, so it's not linearly passive.
That's true.
Propagation from the dendrite to the soma. So all the models are based on linearly propagation from the dendrite to the soma.
That's true. Yeah, that's a good point. Actually, the general question is, if it's hot, or weakly hot, why doesn't the action potential start there?
Not really.
No, but this is the question, really. Because if it
is
is
a question.
a question, you ask another question, but this is the preliminary question is, why does it always start here if this is already nonlinear there? Because it has nonlinear channels, so why doesn't it start there? That's one aspect. And the answer is that it's, so it's a deep question, because you can see that it backpropagates. So these channels are coming into action. They do participate, these channels, in the backpropagation, in the boosting of the backpropagation. So why don't they, or maybe they do, participate in boosting the forward synaptic potential? That's what you asked. Yes? Why don't, if it is nonlinear, if the dendrites are nonlinear, maybe the synaptic input is boosted, the synaptic input is boosted by these same channels, yes?
Okay, but
That's the question.
It can plug into the integral and fire model. Because you can say the nonlinearity is summed in the axoniote or something.
Yeah. No, but your question was right. I have nonlinear devices here. I have nonlinear ion channels there. It could be that the dendrites themselves, the input to the dendrite is already feeling, is already experienced nonlinearity. The input itself, the synaptic input is being boosted in a nonlinear way because you have these channels there. So can I assume that the dendrites are passive this direction and active this direction in some sense? So the answer is almost yes, because the synaptic input is relatively not so big, not so large, the synaptic input itself. Unless it is on spines, and spines has different channels, but locally, the success of this backpropagation to activate these channels is because you start with something very big here, 100 millivolt spike. You start with 100 millivolt spike. So this 100 millivolt spike, it's big enough to be able to really recruit channels here. And it's big enough to recruit channels there. So if you start with a big signal, if I would start with a big signal in the dendrite, if I will inject to the dendrite 100 millivolts, then I will activate, of course, the channels do not know if it's coming from here to here. They just see 100 millivolt or 50 millivolt or 60 millivolt. They don't care if it comes from here or there. The channels do not know direction. They know the local voltage. So I'm saying that the synaptic input is not big enough to really boost in a significant way, so it's almost passive. Not exactly, but it's almost passive. But it's certainly not passive this direction, as I show you here, but it is mostly passive this direction, but not really completely. But it's a good point.
So these channels have a higher threshold than the ones that generate the action?
They also have higher channel, and because, not that they have higher channels, but because they are less dense, in order to really start a regenerative phenomena, so self-activation, you need a higher threshold to have enough of them to inject enough current in order to locally boost. So if the channel is a channel and it's activated at a given location. So the channel has the same threshold everywhere if it's the same channel. But in order to start, so one channel cannot start the process. There has to be enough of them so that the current that is generated locally, some of it escapes axially, but some of it needs to reboost. So for example, in the axon, if I will reduce the density of the channels here, by TTX, let's say, but not block, just reduce, I will need more voltage to start the spike. So the voltage threshold depends on the density of the channels. The voltage threshold, the less dense channels I have, although it's the same channels, the more voltage locally I will need in order to recruit enough of them to overcome the axial loss of current. Because this channel that inject current, a lot of it goes sideways, and it doesn't succeed to boost locally the spike. So you need enough of them, so there will be enough local charge to boost the channel and generate a full spike. So threshold depends on density as a collective phenomenon. Threshold is a collective phenomenon, not of a single channel, but as a group of channels. So that's why threshold here is larger, because of the density, not because of the different channel type. Okay, so I just wanted to tell you that because the question was very interesting question was raised whether there is some other signal that may tell distal synapses that they did something good, so to speak, and generate a spike, we found, or these people found, this is also Stewart, this is Sackman, but with Matthew Larcombe, they found that this long dendrite, could be 2 millimeter long, has another initiation spot here, hot spot, and this is the hot calcium region. So there are in this place, there is this hot calcium-dependent voltage channels, not sodium, calcium-dependent, that may generate local spike, only local spike, not propagating spike. Local spike that looks like this. So you see the calcium spike, which is initiated in this region. And we believe, and people have shown, that this is now a local signal for all these synapses if they cooperated and succeeded together to generate this calcium spike, which then, of course, boosts. It doesn't propagate, but it generates a lot of inward current because it's a very broad spike. It's a very broad spike. It generates a lot of extra inward current, which generate extra sodium spikes. So these are sodium spikes at the cell body that are the result of the calcium spike at the dendrite. And so this would be a good way to signal these synapses you acted together well enough, you generate calcium spike. This calcium spike added a sodium spike, so it means that you should become stronger, let's say. Spike timing dependent plasticity. But this spike timing dependent plasticity depends on the calcium spike. And the calcium spike generates additional sodium spike. Is it clear? So this is an extra kind of a very interesting new signal. We did not know about these signals before because we could not record from there. Now we can impale with an electrode and see what are the conditions to get this boosted calcium spike.
How does it know that it had the sodium spike again?
So I'm showing you. Okay, so this is the experiment, which is an interesting experiment by Matthew Larcom and Sackman. So if you inject to the cell body a little short stimulus, you get sodium spike, the regular sodium spike that you saw before, and this sodium spike backpropagates. Okay, so from here it backpropagates in the red region. Let's say you see this sodium spike. By the way, in the red region you see this little hump. This is the beginning of another nonlinearity, which is the calcium-dependent nonlinearity, this little hump. But it's not full spike. It's just a little boosting locally. But if you do the following experiment, which is in this case timing local synaptic input, so they stimulate the red stimulation by the electrode, gives you locally here a little simulated EPSP, artificial EPSP, because you injected current here, and you get local EPSP, let's say here, on its own. If you only stimulate the cell body, you get this phenomena on its own. But when you stimulate both of them in the correct timing, so you stimulate first this one here at the cell body, you stimulate and you get the first action potential at the cell body, and a little bit later on, you stimulate your electrode here, an artificial synaptic input, both together, you see suddenly that there is the backpropagation action potential plus your synaptic input together generates this calcium spike, this red spike locally, and as a result of this red spike at the cell body, you see these two extra spikes, these two extra sodium spikes at the cell body. So these two extra spikes are not the result of the stimulation at the cell body, because the cell body just stimulated once and you got this spike. These two extra spikes are the result of this big action potential that is happening locally there. But you can also do it if you stimulate very strong here, stronger than here. Okay, so you get a big local EPSP from big strong stimulation here. You can get on your own. You can stimulate local calcium spike here, and again you get these two extra sodium spike at the cell body, or the axon. So you can see that there is this kind of a ping-pong effect. You can call it a ping-pong effect between the local calcium spike that usually would happen either if you get a very powerful synaptic input here, or if you get a coincidence between small synaptic input here plus backpropagating action potential. This coincidence detection locally will generate a calcium spike, and it will generate extra sodium spikes. So this will be the output of the cell body. And you know, Larcom and Sackman has a whole theory about the fact when you see a tack, 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 it will tell you, in general, that there was a coincidence between regional input here, let's say to the basal dendrite, input there, and the tack, tack, tack is a signature of a coincidence bottom up and top down. There is a whole several papers theoretical about how do I connect top-down information, you know, because this dendrite receives top information from other distal regions are converging into this into this region. So this is top information. Let's say if this is V1, it could come from V2. So some a priori knowledge, top down, coming here. Bottom up from the thalamus mostly comes here. So the theory was, their theory is that this coincidence of some information coming from the outside, let's say a tree, so to speak, a tree, a face. And there is some a priori knowledge or assumption that this might be a tree or a face. If I have this coincidence and I have tack, 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 it's a confirmation that there is the coincidence or agreement between my top down and the bottom up. something like that. I don't have time to talk about this theory, which is very interesting, because there is this issue, how do you really navigate stream of information coming from the outside and some a priori information coming from upper levels that are converging here? How do I merge them to have a specific signal that tells me that there was, so to speak, an agreement or some kind of a correct coincidence between what is coming here and what is coming here. It's a general theory. You may look at it later on if you want. Maybe I should have given you this as a paper because it just came out as a review. But this just to tell you that people are trying to give a meaning to such a signal, but a functional meaning to such a calcium signal, or to this tak-tak-tak, which is rather rare to see in neurons unless you get this kind of coincidence. But you can get this tak-tak-tak, as I said, by very powerful input on its own. So you may ask, how does the neuron know that it's from this? Well, in this case, you have 3 spikes. In this case, you have 2 spikes. Maybe it's a different signature or not different signature, but that's what I really wanted to tell you, that there is 2 things. I wanted you to not always to think about a spike as an output signal, because I show you a local spike here, which eventually becomes output, but by another signal, which is the sodium spike. I'm just showing you an active, full action potential, not a small nonlinearity, huge nonlinearity, a very huge nonlinearity, the local calcium spike, which is somehow a local signature. We think it is mostly talking to these synapses, yes, sensitive to the input here, and it conveys an output eventually there, and it could be also a plastic signal, meaning that if you have a calcium spike and these synapses work, so I have this spike time-independent plasticity mechanism maybe, but not due to the sodium spike propagating, but due to the calcium spike seen by these synapses somehow. Yes.
Can this kind of calcium spike come only due to the dendritic activation? So it will be like
here.
coincidence?
Like here.
Yes,
Here it starts only due to the...
Yes, but here you artificially stimulated it very strongly.
Yes, or you ask whether in real life, when I'm talking to you now, do I have calcium spike in my dendrites? I mean, without... Only due to dendritic tree. It's of course not easy an experiment to do in vivo. In vivo. First of all, we see calcium spikes in vivo because we have this, 2-photon imaging techniques and also calcium imaging techniques, so we can really see these calcium spikes. And this is very beautiful work by Helmchen showing calcium spike in vivo while the animal is whisking or doing other things in the visual cortex here. So you do see these nonlinearities, but whether it comes or not with a sodium spike, that's more difficult because you have to at the same time see that it depends. And so this is another imaging because, or recording at the cell body and showing that this calcium spike always happens, yes or no. I don't know. I don't know myself whether there is a conclusive answer to your question. But the idea is nice. Yes, the idea is nice, really. That first of all, the fact that you have a local calcium is interesting. That there is this huge spike. And the issue that this local calcium spikes is favorably starting when you have this backpropagation plus weak, semi-weak forward stimulation is also interesting here. But whether it has a direct functional implications, that depends. That's the theoretical part. We don't really know.
2 questions. First, don't we have chemical signals which would be sufficient?
Chemical signals to do what?
To the distal branches.
What do you mean chemical? What do you mean, for example? What signal? What signal do you look for?
Chemicals.
Some chemicals that go through the dendrite. Some chemical. Could be. There are a lot of chemicals, of course, running all over. If you think, for example, about ions, which are also chemicals, for example, calcium can flow inside, and there are waves of calcium in dendrite. Waves of calcium. Not only of voltage, but of calcium. So these are the type of signals you are looking for as signals flowing in the dendrites, saying things, calcium, for example.
To signal the distal branches?
They may be sufficient, maybe, but they need to be generated somehow. And in this case, in this case, well, again, it's very difficult to know. When I'm telling you that there is this calcium spike, and of course, with this calcium spike, there is a lot of calcium flow into the cell. So if I would image calcium here, I would see, bursting of calcium locally and also spreading of calcium, and people show it. Whether it's the calcium that flows, the signal for plasticity, or whether it's the voltage itself without the calcium. But they go together, yes? They go together. In this case, they go together. So what I need to do, and people started to do it, is to inject myself a voltage that looks like this, let's say, yes? Somehow blocking the calcium channels. Again, like we did before, blocking the sodium channel. I'm going to block the calcium channels, have a voltage look like this, and ask whether I get plasticity or not. So I separate these two parameters that, in this case, go together, because the calcium spike is depending on calcium. So you see calcium. So I don't know to answer whether calcium enough is enough. The other way is just to inject calcium without voltage. So this will be the other, closing the loop. So I don't know the answer. I don't know to separate today. You cannot say it's only voltage. Mostly people think that it's the voltage that really affects the NMDA receptors, because NMDA receptors are involved with plasticity, and they are voltage dependent, the NMDA receptors. So people think that voltage itself is a significant, maybe not the only one, but important one, a signal, because it is intervening with the opening of the NMDA receptors, and this is initiating the plasticity.
The second question, that tac-tac-tac theory.
That tac-tac-tac theory is a good name for it, yeah?
Does it mean
Tac-tac-tac, yes.
if you artificially generate tac-tac-tac voltage, they have plasticity changes?
No, because this tac-tac-tac is not propagating to this location. So the sodium spike, as I said, the sodium spike itself cannot propagate so long, so far. So the fact that I have a tac-tac-tac in the cell body, these synapses do not know the tac-tac-tac, but they know that they generate calcium spike. So I'm saying that from the synapse point of view, the teacher is the calcium spike. Let's call him the teacher, the plasticity teacher.
The tac-tac-tac is only
Is for or side effect or computationally important. I'm trying to I'm saying two things here. There is an issue of plasticity, and there is the issue of coincidence detection. The tac-tac-tac. The tac-tac-tac could be not for plasticity. The tac-tac-tac could be for coincidence of inputs. Not talking about plasticity for a second. And the calcium spike could be, sorry?
the tac-tac-tac is signal for the stoma?
For the output of the cell. The cell output is tac-tac-tac in this condition. And this tac-tac-tac could signify a coincidence between input here and input here. That's one thing.
The signal for the sinus is sodium calcium spike.
And for plasticity, and this was shown, that for plasticity, for these synapses to change their efficacy, it's not the sodium spike, because you can block the sodium spike by local TTX. People spread local TTX here. He will be here in April, Spruston, who did it, and will give a special seminar. You can block that by TTX, so you know that there is no back propagation, for sure. Not only no back propagation, there is no sodium spike at all. But you can generate this calcium spike with synaptic input, and you see that this is a teacher on its own. So there are two teachers at least. This sodium spike is a teacher for these synapses, maybe. This calcium spike is a teacher for these synapses, but they interact, these two teachers. So you have two classes, yes? I'm a teacher, I'm the calcium teacher for this class, he's the sodium teacher for this class, but we interact. We interact, because if I'm a teacher of calcium, so I affect the other teacher, because I generate two tac-tac, yes? So it's a classes that interfere with each other. Somehow the synapses have to learn together with two teachers. So that's just to highlight the fact that that these neurons are very interesting nonlinear devices that are both plastic but also coincidence detection of some inputs that are in a distributed system, of course, you cannot have it when you have a point neuron, this kind of nonlinearity. Okay, because I want to speak a little bit more about, let me see. I'll do 5 minutes just highlight of things that I'm doing now, but I don't want to speak about myself in this course now. I just want to tell you a little bit one word, because you will hear so much about this blue brain project, the human brain project, the €1 milliard that we got, all these things. I just want to very fast to tell you one word about that, not to tell you the whole story of the human brain project and why Obama suddenly decided to put $3 million on this. A very interesting, by the way, project that they are trying to develop. They call it the BAM project, the Brain Activity project, or mission or something, but brain activity, because they want to develop in America techniques, new technologies based on molecular biology and nanotechnology for recording from above 1 million cells simultaneously. Today we cannot record from more than 1,000 with 2-photon imaging with electrodeless. So this BAM mission of Obama, maybe we'll tell you about this in Binyanei Haoma very soon, who knows, is really an American project. Our project is something else, which is related, of course, because brain is brain, you know. And so we are trying to do some kind of a systematic analysis of structures in the brain, both morphometrically, just to know who are the characters in the brain, how many cell types, who are the cell types in the brain. There are many types of types. You can speak about morphological types in terms of, you know, I say pyramidal cell is a morphological type. Say spiny cell is a morphological type because it has morphology, so I use morphology to nomenclature, as a nomenclature, but I can speak about electrical types, and I can speak about molecular types, and I can speak about chemical types. And people speak about all these things, but there is no kind of an agreement over how many players in terms of types, not in terms of number, how many players, types, you have in the cortex, for example. So that's one aspect, just to know who are the characters, just to show you that what came out of this project up until now is that in the cortex, in terms of morphological types, you have about 60 morphological types. For example, you look at layer 1 in the cortex, and that's one of the results of the Blue Brain project, is to really systematically database, we can call it the databasing of the brain at the level of cells in this case. You can see that, for example, layer 1 has only inhibitory neurons. And these are the different types. So I don't want to start with the names. So layer 2, 3 have this type of inhibitory neurons and this type of excitatory neuron and so on. As you go deeper into the layer, let's say layer 5 already has several pyramidal cell, spiny stellate cells and other types, excitatory and inhibitory. But you can see an interesting phenomena that you may not have known, that in terms of types, not numbers, in terms of types, you see much more inhibitory types than excitatory types in the cortex. Although in terms of percentage, you get maybe between 10 to 15% if you just count. So you inhibitory cells. So there is much less number of inhibitory cells, but much more types of inhibitory cells, in this case morphological types. Also electrophysiological types, because I can characterize the types in different ways, as I said, and I can record from this cell and from this cell, and I see that this is a burster, and this is a regular firer, and this is a stutterer, and this is an accommodating cell. So there are different types of firing types. And this is another work that we did using cluster analysis objectively. All this, by the way, is an objective. You throw the different cells into your algorithm, and it gives you the types. It's not that you tell them that this is a type. They have to objectively say that this is one type of tree, this is another type of tree. Is it type by layers or type not by layers? All this is a systematic classification project that's part of the, and this is morphologically. There is another thing that we did here. I did with Shaul Druckmann, who was a student at the ICNC, using cluster analysis technology to classify electrical types and so on. So that's part of the project. So just systematically put all these things. Eventually, of course, you want to understand what is the significant, what is the functional significance of having all these types. Because I can say, okay, all my inhibition is inhibition. Okay, there is one inhibition, there is one excitation, and that's enough. But that's not what happens, that's the thing. It could be functionally that you have just one type, and that's the most basic classification of neurons functionally. There is a functional type that is inhibitory, there is a functional type that is excitatory, and there is this interaction between them, inhibitory to inhibitory, excitatory to excitatory, and so on. Okay, you can compute certain things with it. But apparently the cortex is built differently in some sense. We don't understand it. Not only the fact that you have so many, why do you put so much molecular energy, so to speak, to create all these types? And you know, each type is making contacts to subset of cells. So this inhibitory cell connects to this, this, and that. And this inhibitory cell connects to this, and that. So you have all this kind of local, kind of non-random connectivity that eventually gives you a cortex. And we don't understand it. Completely not understand it. So that's part of the project. So I'm not going to talk about this project, just to give you that we decided eventually to really dwell into, in this case, classification, and also molecular classification, and eventually get a good kind of model, both electrically and morphologically. So really, in terms of types, in terms of percentage of inhibition per layer, the different types of cell per layer, and so on, and eventually get a column representation at this level of resolution, yes? And as I said, you want also to characterize the types of firing objectively, and this was already done. We know that there are about 14 types of electrical characters. One is doing this ta-ta-ta-ta-ta, but this is an adapting cell, and one is a burster, ta-ta, stop, ta-ta-ta-ta, stop. Each one has its own kind of machinery for firing for some reason. So they have all this kind of interesting language, mostly the inhibitory cells. Excitatory cells, which are the output cells, the long-range projection cells, are more kind of boring, although they send the information out. But the local circuit, which is inhibitory, mostly the interneurons, that's why they call the interneurons, the local neurons, are rich in their firing repertoire. They are rich in their morphology. So the designer decided to put a lot of efforts in generating a small population. Let's call them brakes. The brakes, the inhibiting population. They can play a lot with all their kind of machinery, tak-tak-tak, tak-tak-tak, and this particular structure. And I also mentioned last time that it also occupies a particular dendritic region. So this tak-tak-tak type, let's say the basket cell that sits here, only talk to this region of this pyramidal cell. So there is this kind of local geometry or local strategy, a local strategy to generate circuits with all this richness of activity and also of morphology that eventually gives the system a very rich output dynamics, which we don't understand. We really don't. So why did the designer decided, maybe it's very clever, you want output to be very rich, you want to talk to different regions far away with a rich repertoire of outputs, and instead of putting all the energy on the 80% output cells, you put a lot of energy in the 20% or 15% local cells that can manipulate manipulate in a sense of activity, and shape, sculpt. They sculpt the local activity that will eventually be heard outside. Maybe it's just economically very efficient. Yes, instead of putting my energy on the output, I put my energy on the local control from an engineering designing point. But we don't understand it. I'm just throwing ideas, just based on what I just showed you. There are a lot of inhibitory neuron types, not numbers, and a lot of channels, different channels expression, so different firing expression in these particular types. I don't know why. It's an open question. Okay, so this is part of the human brain project, but I'm not going to tell you about that much. We are going to the computational level because that's what I really wanted. Just to tell you, in a second, I'll give you the time to answer. As I said, there is also now a big effort to try to characterize, because we have all these molecular tools, to characterize cell types based on their gene expression. Because eventually, the gene expression is what really determines everything. It determines the shape. It determines the type of synapses. It determines the firing channel type protein. So the genes are really, really the underlying substrate for generating types. So there is an effort today, this is not in Israel, this is in America, by a group of cells to really start to really say what molecular or genetic types of cells do I have, let's say, in the cortex. Do I have 200 of them? Do I have, I don't know, thousands of cell types genetically in a normal brain? I'm not talking about the sick brain, of course, and so on. And so this is going to be done, I'm sure, within a year or two, both in Allen Institute, but also in other places where they really take, in many techniques, they really take the inside of the cell, so to speak, and characterize the expression of different ion channels using single-cell PCR and other techniques that I don't know much about them. Okay, so ask a question or two, and then we'll take a little break, and then we'll go to this. Yes?
Do you see the same types of cells across animals? Because it says the human brain product, but I guess it's mostly not from human.
Not from human. Not from human. Okay, so that's first. Okay, so I'm not a genetic person, and it's very hard for me to even answer your question. Although people did work on that, if I take layer 5 pyramidal cell from human, and I have these cells today, fresh cells from human, layer 5 pyramidal cell, operation, after operation. So we have today, I'm working with a group in Amsterdam where they are in hospital. They work in the hospital whenever there is an operation for, let's say, a tumor, let's say in the hippocampus. You have to take a huge piece of cortex, I mean huge, a whole piece of cortex like this, which is bigger than the mouse brain. Take a whole piece of human brain, and then you can ask questions like this, genes, types, morphology, electrophysiology. So it's starting. No, they take it out. Usually the physician in the medical school here, yes, they throw it. Well, what can you do with the human? They throw it to the garbage. What do they do with a human piece? So you don't bring it back to the brain, yes? But in this case, in Amsterdam, they take it directly to the lab, immediately. So they do genes, and they can start to answer what is the difference between gene expression, let's say in layer 5 pyramidal cell in frontal cortex of human, and whatever relevant piece of brain. Even between rat, these are not different, not exactly the same genes in frontal cortex or temporal cortex of the rat and the occipital cortex, and so it's not exactly the same genes, but within a given region, I can say, okay, I'm going to do V1 of human, and I'm going to V1 of rat or mouse, and I want to compare the cell types and so on. So I can already tell you that in humans there is no 6 layers, but 8 in the frontal cortex. 8 layers. We are not rats. And also, also different cell types in some sense, but still there is layer 5 pyramidal cell, there is layer 2-3 pyramidal cell. Are they the same in terms of gene expression or not? I don't know. I don't know to say today, but it's coming. It's coming.
So why is it called the Human Brain Project and not the
Because that eventually, of course, we want to go there. Eventually, we want to really characterize what makes us human in terms of cell types, in terms of, for example, if I just learned from being in Amsterdam, they succeeded to completely characterize the channel, the receptor types between layer 5 to layer 5 pyramidal cells, so the synaptics. And it's extremely close in terms of the synapse between the mouse and the human. The synapse itself, the NMDA receptors, AMPA receptors, and all other receptors and channels and proteins in the synapse are extremely similar. That's why medication works so well when you develop it on mouse, and then you take it in human, because of the synapses. So we are very close, but apparently not enough close, because I can speak here and the mouse cannot. So there must be something different, or the chimpanzee cannot. So there must be different, but we don't know enough about this. So the Human Brain Project is exactly going towards this, building along the development of techniques in the lower, in the animals, but using the same techniques then whenever you have a tissue, which is more rare, all right. So that's one aspect. There is also a project in Germany, Amsterdam, and other places where they have this human bank, dead brains. So, and the Allen Institute as well in America, Paul Allen Institute, where they take people that donated their brain after a car accident, and then so this is dead tissue. This is dead tissue, unlike what I said before. And then there is a lot of anatomical work about regions, about numbers of cells, which you can do also. And also you can do a lot of molecular biology on dead tissue. So for example, the Allen Institute, which is the most successful institute, succeeded using dead tissue to characterize which genes are expressed in what areas in the brain. First of all, how many of our total genes, the 3 billion pairs or the 30,000 genes we have, how many of them are expressed in the brain of humans, male versus female, and now also sick brain, let's say Alzheimer's sick versus not, depending what brain do you have. So all this can be done on dead tissue. But if you want to understand dynamics of a system, of course, you need spikes, you need synaptic activity, you need spike timing dependent plasticity. So there is now a new work on two cells, whether you have STDP. For example, we don't know if there is backpropagating action potential in human cells. We believe that there is, but nobody recorded from dendrites of humans, so I cannot say that there is. Is there a calcium spike in human, layer 5? I don't know. All this has to be done. But I do know that there is STDP because people recorded in Amsterdam from two cells and showed that there is spike timing dependent plasticity. Whether it depends on some other mechanism or backpropagation, we don't know. So it's starting. But it's a very rare material and it's very hard to, but that's the direction, because eventually, as I said, we want to develop medicine directly to the brain of humans, not only of rats. So that's the project. So let's take 10 minutes break and we'll go a little bit to talk about dendritic computation.
צריך שהם מאריכים את העבודה בעוד 6 שבועות? לא נותנים לי בכלל לעבוד. אז זהו, שלא, כתוב מבודד, לא יכול להיות ביום חמישי, לא יכול להיות ביום חמישי אחד, לא יכול להיות שעה טובה. זה חייב להיות יום, מה אני צריכה לעשות? תקווה, ביום האחרון. שבת תודה.
מה, גם מבחינה בירוקרטית, או שהם פשוט לא רוצים
לא,
הרקע,
נראה לי שזה מבחינה בירוקרטית, ועם זמן מאוד הם שמחים שזה להמשיך ולפחות להפעיל אותי בטלפון.
The two models that have been proven
אני לא אדווח על
physiologically,
חצי
or
שעה.
am I,
No,
you know the model that is also,
no. So it's a very different type of model. The Hodgkin-Huxley is a mechanistic model at the level of single cell. So it was. So I think it's different models in the sense that the Hodgkin-Huxley came directly from the data. So there were data, they recorded spikes, they recorded currents and all that, and eventually they built bottom up. They built a model based on data. Here in the Rafa case, there was a phenomenon. There is a phenomenon of direction selectivity. And so there was a perceptual kind of model. There was a conceptual model. Like Hodgkin-Huxley, which is a mechanistic model. Here there was a conceptual model that there is this, delay lines and all that. I will speak in a second about that. And then they were looking. So it's a reverse kind of order. But eventually, I can say that this is very recent. That now, again, there are some caveats, and it's not completely done. But there is a progress in terms of the conceptual model of Reichert being, they found the basis, the anatomical and physiological basis for this conceptual model. Hodgkin-Huxley did not have a conceptual model.
okay.
They saw the spike and they built the model for it. So it's a little bit different kind of model types. But also, if you speak about bottom-up models, I think the model of Rome, the cable model, the cable theory, the change in shape of EPSPs, the attenuation, all these basic things that I taught you, was now recorded, because you can record the synaptic input locally, you can record it to the cell body, and you can see how much it was attenuated, did it change the shape. So many of the cable predictions, the predictions of cable theory, which was a prediction because we could not record from the dendrite then, are now confirmed. firmly. So in that sense, I think both Hodgkin-Huxley and Cable's theory for dendrite are solid in terms of theory and models. And the Reichardt is at a different level that is starting very happily to prove that some conceptual models are found in the tissue.
And then so the main success of a model is how much prediction.
So that's the ultimate not only how much it can replicate the phenomena and what does it predict new that you would not think about this without the model. Right? The model should direct you in some sense to do experiments that you would not think if you didn't have the model. So yes, the meaning of success is like that, but it's very, very, very sparse, unfortunately. Okay. So actually, the whole talk, the whole 3 hours or the whole 3 meetings between us was actually to try and focus on this. I don't have much time for it, but it's really not important because the next 3 papers that will be represented or presented by the 3 of you on this coming Wednesday will be around computing with dendrites. So let me give you just a very kind of abstract thing about this, and then we'll hear next week. Okay. So there were several ideas around dendrites, and I don't want to mention all of them in a specific way or in an elaborated way, just to tell you that a very simple idea, of course, was that because you have a distributed system, you can do locally, you can speak about subunits or regional or domains of dendrites and think about each domain as a local computing device or a local plastic device. So things that are happening in this part of the dendrite and in this part of the dendrite, both plastically but also non-linearly, could be locally be done first and then only later be heard or summed at the solid body. So this issue of subunits, that again, of course, if you had a point neuron, there was only one local subunit. But thinking about dendrite as distributed subunits, where in each subunit they act like a local computing device in some sense, nonlinear computing device. I will talk a little bit about directional selectivity because this was one of the original ideas that dendrites can compute something in a very specific way. I will show you the role idea about directional selectivity, and then we'll talk a little bit about more recent ideas about directional selectivity. If I have time, I will show you that when you have 2 compartments, you already just heard now, that when you have 2 compartments or distributed system with 2 compartments or more that are nonlinear, the compartments, and there is interaction between these 2 compartments, you may get repertoire of firing just because of this ping-pong effect between these 2 nonlinear regimes, this enriching the repertoire of firing without adding more channels, but just having these 2 distributed spatially and temporally regions, you may get interesting firing repertoire without having additional channel types, so to speak. Okay, so there are other ideas that again, I don't want to go into it because of time limitation, but there were along the years some ideas, and as I told you recently, because we succeeded, I started the lecture by saying that people succeeded today to recode both locally from dendrites, like this fantastic work by Arthur Connorth, showing that the input itself has a directional selectivity and the output may not have the same directional selectivity. Something in the dendrites is computing eventually which will be the dominant directional selectivity. How this computation is implemented, in this case, is not yet clear. In the case that I showed you before, and recently I mentioned also the paper of Jackie Schiller from the Technion, being able to, from the inside, block certain type of nonlinearity of one single cell, or maybe I didn't mention it. It's a continuation, actually, of the work of Arthur Connorth in some sense, and she takes the directional selectivity of whisker cells. So you can move the whisker this direction, this direction, this direction, this direction, and record from the appropriate barrel representing this whisker, and you can see the directionality, the response of the cell to which is the preferred directionality. But in this case, it's not visual directionality, but whisker directionality. She could block from the inside of the cell by injecting a drug into the cell. She blocked the NMDA nonlinearity, showing how much of the directional selectivity of the cell is determined by the nonlinearity of the NMDA receptors. And she showed that there is a bias. So a given cell has a bias to prefer a direction, let's say 45 degrees of movement, and these NMDA receptors really accentuate, they make it much, much more to this direction and relative to other directions. So it's a boosting. The already biased direction is being boosted by the nonlinearity, which makes it very, very obvious that this is the direction and not this other direction. So that's a very nice work. on this. But let me start just with the history. So this is this dramatic fellow that you should have heard about, and this is the book he read. So this is McCulloch, or from McCulloch and Pitts. So these two characters, very, very important and very, very interesting fellows in MIT, wrote this very important paper, a logical calculus of the, that's a very nice name also, title, a logical calculus of the ideas immanent in nervous activity. So they took advantage of the fact that a neuron is all or none, not all or none, and they used this binary properties, the 0-1 property of neuron, to write a whole heavily mathematical paper from 43 on what kind of computation can a system do when it has a binary elements with 0, 1, yes? But not only that, but also an element that has, the input has two types. They have the excitatory input that tries to go into the 1, to excite the cell, and there is another input, the inhibitory input, that tries to veto the output. So they use these notions from neuroscience to develop a whole theory that actually pushed computer science or development of computers. You know, MIT at this time, there were several very important theoreticians developing computational theory, computers, and they were one of the, so just for you to understand that the brain, simple ideas about the brain pushed the computers. This paper did not impress very much neuroscientists. Up until today, neuroscientists are not very impressed with theoreticians. Yes. But anyway, McCulloch and Pitts neuron is a very classical kind of mathematical unit, if you want, or logical unit. So this is the M and P neuron, the McCulloch and Pitts point neuron. And you see this point neuron receives excitatory synapses. It may receive inhibitory synapse. And because it has this 0-1 output states, it can do an operation like this with this kind, assuming, let's say, just for the discussion, assuming that each input can generate an output, let's say, but not if you have inhibition. So you can write this logical sentence. You can say E1 or E2 or E3 will generate an output and not I. So this is a logical sentence. Okay, so that's another, I'm just assuming here, just for the demonstration purpose, that this could be written, this unit could be implemented, this logical operation, assuming that each one on its own. If I would need two excitatory synapses to reach threshold, let's say, then I would write E1 and E2, or E1 and E3, or E2 and E3, to synaptic combination, and not I1. Let's say that I is so strong that it can kill. Of course, this is a very simplified notion, because we know that inhibition is not completely vetoing, it's not killing the excitation, it's graded. So this is a very crude approximation. I'm just showing you that because cells were known to have 0-1, spike or not spike, and because cells were known to have excitation inhibition, McCulloch and Pitts used this to develop a whole mathematical theory about what kind of a universal machine you can have with units like this. What kind of computation can you do with units like this? Okay, and this gave a lot of push to development of modern computers, because you know that there you have the 0-1 states and so on. So this was very, very important paper for the development of computer theory, computational theory. But of course, we don't think that neurons are logical devices necessarily. But this was taken later on by other people like Koch and Poggio to say, okay, so what kind of operation can I do if I have dendritic tree? Okay, not a point neuron, but a distributed neuron. For example, suppose I had, let's say, this dendritic tree with this architecture of synapses, the red one are inhibitory, and so on, then I can write another kind of logical sentence, logical operation. Let's look at it for example, this case. Because there is now an importance of the relative location of inhibition to excitation, this inhibition can veto, let's say, this inhibition can veto everything that is coming from here. But this inhibition can veto only the more distal excitation. So this inhibition cannot affect this excitation from the cell body point of view. So this is what is called the on-path condition. And this is actually true. So if you want to really have a very global inhibition, you should put your inhibitory input close to the output, let's say to the axon. If you want to have a very local inhibition that will affect only locally, so this subunit, this local operation, this inhibition will veto this excitation, but not this excitation, because it's not on the path between excitation to the source. So this is the on-the-path condition. Is because of this on-the-path condition, there are these different logical operations depending on the geometry of the connection. So this is a nice idea, that just the geometry of the connection and this on-path condition will determine the logical operation, for example, yes? So here, let's say, it will be E3 and not. This is a mistake here. And not I3, I2, and I1, okay? Because if this is, let's say that a single synapse can activate a spike, you want to activate this, but not this three type of inhibition. And if it's E2, it will be E2 and not I2 and I1 and so on. No, it's not written well here. And eventually, if it's E1, it's E1 and not I1, because this I1 only affects this one. So anyway, so this is the kind of operation. Just to give you an idea that this idea of Michalak-Pitt was pushed a little bit further by Koch and Poggio to show that dendrites with their synapses can implement complicated logical sentences. The other interesting idea came from the fact that in the retina, as I already mentioned, I think, at some point, and you know well, at the retina of many species, including humans, monkeys, and even simpler, like rabbits and other animals, you have these directional selective ganglion retinal cells in the retina. So you can have a directional selectivity cells. For example, if you record from this particular cell, so this is a intracellular recording from a ganglion cells in the retina, and you show this, so to speak, ganglion cell. So this is an isolated retina. Take the retina out, and you show light to this retina. Yes, you move bars in different directions, and the retina, so to speak, sees these. You can see that in some angles there is just EPSP, and suddenly in some angles of visual angles, you suddenly see an output. So you will say, this is a ganglion cell that is directional selective, because it generates output only in this regime of orientation in the world. And you can characterize it with a kind of polar plot. You know, you can look at the EPSPs. You can see that the EPSPs start to grow. You get more excitation. You start to grow in this angle, and eventually you get a preferred direction. You can do polar plot using spikes, and of course it's more sharp because there is no spikes here, spikes, spikes, no spikes, and so on. So this is directional selective ganglion cell. This has to be somehow computed in the retina, because as I said, it's an isolated retina. So the network, or somehow somebody makes this neuron directional selective. The question is how. And this is the first idea that came from Roll. This is Roll 64, where he showed a very simple principle, but very fundamental. And this is the following. And actually, you should already know the principle because we spoke about delays in dendrites. And this delay due to the dendritic cable. So this is a cable that we already discussed. This is the cell body. Synapses, in this case, only excitatory synapses, no inhibitory synapses in this model. It's a model. So these are the excitatory synapses. In each compartment, you have synapses, excitatory synapses. And so you can, the only difference between these two conditions is the order of activation of these excitatory synapses. For example, in this case, you start with the proximal synapses near the cell body in time. So you do tac, this is not spikes, yes? Synaptic input. Tac, 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 tac. From cell body out in terms of time. Or you can activate it in the reverse order of time, but the same synapses. but only reverse order in time. You start with the distal one, tuck, 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 tuck. So you use the same, if you want hardware, you use the same machinery, but you activate the synapses along the dendrite in different order, temporal order, in time. Okay? So, and you recorded the cell body. So always you recorded the cell body. This is the output side, let's call it. And then you can see that if you activate the synapses in this direction, let's say from proximal to distal, this direction, you will get, because you activate nearby synapses, you get the EPSP early on. So this is the EPSP coming from the nearby synapses. And then the next EPSP and the next EPSP will ride, so to speak, will summate temporally, temporal summation, but on the decaying phase of the first EPSP. So if there was only the first EPSP, it would look like this, and it would decay. If there was only this. But because you have the more distal one, and it comes later, it rides on the shoulders, if you want to call it like this, on the shoulder of the first EPSP, and the summation looks broad like this. But if you do the opposite direction of activation in time, you see this delay. You see this delay because you started distally. It takes time for it to reach the soma. You see this delay, which you already know. That should be delay on this cable. And then if the timing is correct, if the speed, if the velocity is correct, the tuck, 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 tuck is correct in terms of temporal summation, you may build the voltage which rides one on top of the other. Okay, so this comes in, this starts first, it goes here, then you activate this one, it goes here, then you activate this one, it goes here. So you build up. If the timing is correct, you may build up and get this kind of EPSP. Composite EPSP. It's a composite EPSP because it's for many, many synapses, yes? Is that clear, the little experiment? This is the theoretical experiment. And you can now think about, let's say that you have a threshold here for voltage threshold. Let's say that your threshold is here. Then one direction of activation will fire a spike, this one, and the other one not. So this may become a directional selective neuron just based on the morphology of dendrites and the connectivity of the synapses, something like that. 1 second. So you may think like this. This is schematic, my schematic summary of what I just said, yes? So in the null direction that does not generate an output, you will have this kind of summation, yes, that you just showed before. And in the preferred direction, if the light moves from right to left, and the mapping is like this, let's say these are the receptor cells, and let's say these receptor cells make excitatory synapses on your ganglion cells. So this is your ganglion cell, and these are the input from the receptors, and they are ordered. They map the space in an orderly fashion on the dendrite. Then this direction will not generate a spike. This direction will generate spikes. And you will say, okay, this is, I just show you a mechanism, a possible mechanism to generate a directional selective neuron based on excitatory synapses, mapping from space onto my dendrite, and delay line. That's it. So this was the first idea. How could dendrite be used, so to speak, to generate a function? This is the jump I mentioned in the beginning, that from the mechanism of cables, of delays, of synaptic inputs, of spike output, this mechanistic description, you jump to the level of computation. Yes, I compute the direction of motion, which is, of course, very important for us to know where direction is coming from. So this was one of the original ideas in '64. After the '43 paper of McCulloch and Pitts, this was the first paper trying to connect, let's call it the hardware of neurons, to function. At about the same time, actually a little bit before, there was a conceptual notion by Reichardt. So this is called the Reichardt conceptual model for directional selectivity in the retina in this case, or anywhere. The idea was very simple, actually. There are many variations to that, so I'm just showing you one variant. You have photoreceptors in the world. There is light moving. And the asymmetry, something has to be asymmetrical, yes? Because if this direction causes something and this another direction, there must be some asymmetry in the system, somewhere. Some nonlinearity, some delay, inhibition in one side and not in the other. Some asymmetry should break the symmetry because this is symmetrical, and somehow you see an output that detects the differences. So where is the asymmetry? In this case, the assumption that there was in one direction there is some delay line between the receptor and the output, let's call it the ganglion cell. This is very abstract notion, yes? Okay, so only in one direction you will get. So if your movement is from here, you activate this direction, this path. Because of the delay, it comes together, let's say, and only in this direction you will get an output. Because here you need some kind of, let's say, multiplication or summation. So the delay, because there is a delay between this point to this point, if it matches well to the delay on this line, then when the point moves from here to here, it will activate this path, as you saw, and this will come together, and there will be output. If you activate the input from this direction, yes, from left to right, this will be the first. Okay, it comes here early. The next one will be activated later, and also there is a delay. They don't sum up, and there is no output. That's a very kind of very broad framework. And there are a lot, as I said, a lot of variation to this idea. For example, one variation could be that this is inhibition. Yes, so this inhibits in one direction. It will start here, and it will inhibit the output. But some nonlinearity operation could be through delay, inhibition, and combination of things like that. So I don't want to go into this, but as I said last time, I think to you, and I mentioned very briefly, that one of the beautiful things that came recently, very recently, is this capability to really start to connectomics the retina. So this is this connectomic system that people are now using. In this case, it's Mitya Klofsky who was here giving a seminar. So I asked him to send me this kind of explanation, that you really take the retina and you reconstruct the whole retina. And you try to see whether there are this type of synapses, excitatory, inhibitory, maybe. Maybe there are delay lines, so there is a more direct path from the left than from the right. You are trying now to really go into the system, reconstruct the synapses, reconstruct the how many cells you have between. Really quite an amazing work. I mean, really, really amazing work. If you think about this, until you get some kind of a understanding of a structure, this cell is connected to this cell, not connected, excitatory synapse, inhibitory synapse, and you start to get a circuit description. And ask yourself whether the circuits support the idea. Because I had a very, very kind conceptual idea about what could be delay, what could be generating delays, and what could be making this computation, but is it there? Is it there or not? Okay, so you know that recently two groups actually, in a little bit different ways, describe the following in some sense, but this is a very broad summary of what was found. It was found in this case, particular retina, it was found that this is the ganglion cell. So this is the ganglion cell. This is the directional selective ganglion cell, and these are the beautiful presynaptic inhibitory amacrine cells. This is retina, and we know that amacrine cells are inhibiting, creating inhibitory synapses with the postsynaptic cell, which is the ganglion cell, and people have shown recently, due to this connectomics, the reconstruction, that there is asymmetry in the number of contacts on the left and the right, so to speak, dendrites of these ganglion cells. So in this case, if you get a picture like this, you say, when I move in this direction, okay, there is less inhibition coming from the ganglion cell coming from the amacrine cell to the ganglion cell. Then if I would move the light from this direction, I will get more inhibition from right. and this will cause asymmetry. So this cell will have a preferred direction. If there will be an output, it's likely to come from this direction, not from this direction. So this is one model, recent model of the retina, but this is now based on anatomical facts. It's not anymore just an abstract idea. This is not exactly what I showed you before about delay lines, because this is now asymmetry due to inhibition. But anyway, I don't want to go into it, and we'll discuss it in the next meeting. I just wanted to tell you, because people were really asking, and still today, all this effort to reconstruct the cell at the brain at the level of synapses, which is electron microscope, very hard work to reconstruct this whole thing. It's huge work, terrible technical work, yes, technical work. And you know there is all this problem of identifying synapses and editing whether it's really synapse or not synapse, whether it is from this cell, another cell, all this kind of reconstruction. Huge. It looks very simple, but I can tell you these are many years of work. And so, but eventually it gave something interesting. So we are starting to connect structure to function. And this is the general term. There is a structure. There is a function. And I'm trying to connect the two. So that's the reason for connectomic, if you want. Or at least one reason is to connect structure to function. Okay, just to say a little bit more, just to say just one or two more examples, I told you also that this ping-pong effect between compartments can be proven using simulation, that it could generate the nonlinear interaction between dendritic compartment, which is active, and somatic compartment, versus active somatic compartment similar to that but passive. If you inject to the soma in this case, or if you inject to the soma in this case, you will get different firing pattern at the soma just due to this ping pong. For example, you saw the calcium spike. If I activate strong enough, maybe I will get nonlinear calcium spike, which will go back here and will do this burst, and in here it's not. So Bartlett-Mel, in this case it's actually Pinsky and Rinzel, and also Mel showed that this kind of two compartments or many compartments can give you a very rich repertoire of firing. You can call it dendritic computation or not. And finally, I want to finish with that. That was another nice idea by Bartlett-Mel, and this is the following. He called it the clusteron idea, the cluster, from cluster. Suppose you have a dendritic tree, okay, which is nonlinear. The dendritic tree is nonlinear, so in this case it's NMDA nonlinearity. And you know that NMDA synapses are voltage dependent, as we said before, meaning that if you have more depolarization for the synapse from other synapses, the channel will be more open, and so two synapses that affect each other may be supralinearly summing up. Usually synapses, passive synapses, will sum up sublinearly, yes? One synapse plus the other will give you less than the linear sum of the two. But if you have nonlinearity, like let's say NMDA nonlinearity, two synapses, when they interact through voltage, they may give you more than the linear sum of each separately. So Bartlett might say, okay, suppose I have a dendritic tree, yes? And I have 100 synapses, let's say. Once these synapses are clustered in, let's say, 10 synapses per cluster, this is one cluster, another cluster, and this is one randomly distributed. I activate the same number of synapses as an input. Let's say I have some background activity for each synapse is activated a few times a second, and I'm looking at the output. You can show that because of this nonlinearity, when I'm clustered, I may get more NMDA charge when I'm clustered versus when I'm separated, and I will get more output spikes. So he shows that this kind of capability to have local nonlinearity, in this case supral nonlinearity, you can use it to do very interesting computation. For example, suppose there is a mechanism, let's say, that map my face, my own face, onto the dendrite such that my face, nearby places in my face are nearby places on the dendrite. So let's say my nose goes here. Input from my nose goes to this part of the dendrite. From my ear, let's say here. From my face, I don't know, mouse here. So there is a mapping in terms of localization between my local phase onto the dendrite. And he does it. He's coming from computer science, from computer vision. He showed that in this case, if the mapping is correct, of course, you can classify faces. You say, this is exactly, if the localization is appropriate, you can classify between, let's say, my face and not my face. So not my face will have a little bit different distribution of inputs because it's not exactly coming, and you will get more spikes for my face than from somebody else's face, let's say, or from a face to a car. So this will be a dendritic tree that likes my face, so to speak, and there will be an output that signify my face. This is due to this local nonlinearity. So he calls it the clusteron. So the dendrite become a clusteron. It classifies the input and says this belongs to this classes and this belongs to another class. The question is how many classes can you classify and so on. And this is the clusteron. And Chaim Sompolinsky and Robert came with another idea of the tempotron, which is not only location but also timing of input. So you can use dendrites in some sense to do some interesting computation. Whether it's being done or not, physically now, with my dendritic trees, whether it's really important for some kind of computation, this is the most difficult question because it's not clear how directly to show it, but Jackie Schiller already showed it in vivo. While the system is computing direction of whisker, the dendritic nonlinearity at least enhances or the differences and makes it more extreme to this direction than to this direction. So dendritic nonlinearities should play a role. The structure of dendrites should play a role, and we'll hear more about this in the 3 papers we shall survey on Wednesday at 12 o'clock. So thank you. כן.
הרעיון הוא גם שהקלאסטרים האלה יכולים כאילו להשתנות ברצועה.
בדיוק. זאת הנקודה שהייתי צריך להזכיר, כי הרעיון הוא שהוא כמובן, הוא לא בא מוכן לזהות את הפנים שלי, אבל עם ההסתכלות, הוא טוען, ויש גם הוכחות נורא יפות, שמכיוון שהכל נמצא שם קרוב, בסביבה שלך בתור דנדרית, הכל נמצא שם בסביבה, פיזית. אז עקרונית הייתי יכולה לתפוס את האקסון הזה, לתפוס את האקסון הזה, כי הוא בא בקורלציה, או יש איזושהי סיבה לזה שהם ביחד התחברו לאזור הזה. יכול היה להיות, אין סיבה שלא, מכיוון שהכל בסביבה. השאלה אם אנחנו לא מבינים עדיין, וזה אחד החסרונות הגדולים, אין לנו מודל מתמטי של מה שנקרא structural plasticity. ה-STDP, הרעיון של ה-Spike Time Independent Plasticity, הוא מודל מתמטי של איך הסינפסה הקיימת מתחזקת במפלשת. כן, אז במפלשת יש ל-LTP, יש ל-LTD, כזה Spike Time, אבל כאן הוא אומר בעצם מהו העיקרון של הלמידה המבנית.
השאלה היא מה יותר, כאילו מה יותר חשוב?
לא יותר מה יותר חשוב, אלא מהו כלל הלמידה שלי בליצור קשרים חדשים.
לא, אבל מה שאני שואלת זה עד כמה קשרים חדשים נוצרים או לא נוצרים,
אנחנו יודעים שהם נוצרים. לפחות 50% מהסינפסות שלך במשך תקופה לא ארוכה משתנות. אנחנו קשרים חדשים.
כן, ונוראים שלפני כן לא היו קשורים.
למשל, כן. אני לא מדבר על קשרים, אפשר להגיד, אוקיי, זה קשרים ישנים שהיה להם עוצמה אפס, אבל זה לא, אני אומר פיזית, האקסון שלא היה מחובר. אנחנו רואים ספיין גדל, אנחנו רואים את האקסון עושה ככה, ופתאום הם יוצאים קשר. זה מסה של דברים כאלה, קוראים מסה. אבל לא יודעים מה הכלל. מה פתאום אני מתחיל להתחבר אלייך ולעזוב את ההוא? מהו הכלל? האם זה תלוי בפעילות שלי ושלך, או זה תלוי במשהו סביבתי? אין לנו מודל של structural plasticity. יש לנו מודל של functional plasticity. אז מל אומר, תשמעו, הכל בסביבה. יכול להיות שבגלל שאתה פעלת מהאזור הזה מספיק פעמים, אז האקסונים שהובילו את האינפורמציה מכאן, התחברו שם. האקסונים שעלו מפה, התחברו שם. זאת אומרת, יש לי את התשתית.
בעצם סוג של אותו עיקרון, רק זה אותו דבר בדיוק.
נכון.
Fire together.
כן, fire together, אבל locally.
כן.
fire together, come together, locally. אולי. לא בטוח שזה קיים, אבל זה הרעיון שלו.
בגלל שאת משתמשים את הגבולות, אבל לא
אני נתתי בדיוק באותו אופן. נתתי שיצאו בדיוק בשנתיים האלה.
כן.
זה שיקול, זה חייב את השיקול.
בשביל
אני גם לא חושבת. אני בדיוק אבל כל שאני יודעת יעקב כזאת גאנה שזה נגמר בשיעור אחד, בשיעור הראשון, בשיעור השני לא לדעת.
זה גאטק גם?
לא לדעת שזה משתף שמאל. זה היה את החלפה של התפקידים. כמו בתולבים ברקע. מה אתה רוצה לעשות? בדיוק השיעור הראשון של ההרצאה, כל עבוד סוציאליות, זהו, שיעור אחד.
זהו, אני גם, אני הייתי בשיעור שלה, לא עשיתי את התרגיל. למשל, למדתי פוריה, למדתי ממש קורס סלסה.
רציתי לעשות לך כזה ככה ולא יותר מהבן של
שבוע
כן, ממש הוא דיבר עליו, אבל זה לא היה ממש...
סירב לזה שבאינדיציה,
זה היה גם יכול לעשות את זה.
לא, זה ממש זה, מה שכל הרעיון
זה הדבר הראשון, דברים שאתה אומר אינדיציה, זה
נכון, לא, אני גם צריכה כאילו לא להגיד ממש נאיבי.
האמת היא ש...
לא, האמת ש... אולי לא, אבל... יכול קלט שווה? כן. כאילו, שווה...
מה יש לכל זה?
מלא
בכל מיני מקומות.
לא, זהו, זה לא לומד
הייתי אף פעם לא, אבל רגע, מי כתב את זה?
זה לא משהו בדיוק שזה הבנת עליי. עדיף על הפירוש מזה, לא... לא באמת בדיוק.
זה גם מה שאומר שם, גם בזה עכשיו, גם כל מיני חלקים, בגלל זה כל טוב, זה קורס רק כזה, של 18 מפגשים. והמעברים, אני כסדן רק בצור פוריה, ולא הייתי באינדיאן פוריה. ראיתי את זה ולא הבנתי את המעבר. ממש ממש מסביר את המעבר בין הטור לאינטגרל,
כאילו זה כמו שלסקי חסר
גם מה שהוא אמר, גם מה שאלי אמר עם הקוסינוס, עם הפלוס פי הזה, ואז אתה הופך את זה לקוסינוס סינוס, הוא גם הראה את זה. שם בקורס הזה.
ואז כאילו קוסינוס
סדנו לא נראה לי.
נכון, זה לא
שתיים
מלא דברים, אבל דיפרנציאליות הזה קשה לי מדי. מצד אחד אתה קשה לי, עד שחשבת את זה, חייב לשבת. לא, אני שמתי גבולות.
זה מה ש...
כן, אבל יש לך 4 שורשים. אם יש לך כאילו זה 4, זה I, 4 שורשים.
כאילו לא מקבל את ה...
יש לך בשני קבועים משני הצדדים, אנחנו שם אותם בתור אחד. שני אינטגרלים בעצם. מצד שמאל תעשה אינטגרל לאחד כזה, מצד ימין תעשה אינטגרל לאחד. אז יש שני קבועים בעצם.
אז האמת היא ש...
ואז הם בעצם הופכים לי את הקבוע אחד, כי לא באמת...
לא, אבל בדרך שלא תחליט את עצמו. כאילו, בקצת שלך חלק לעשות שם ב-P0. שזה 10. אז זה לא משנה.
כן.
אז במקרה של P0 זה אותו דבר. אבל אתה צודק, כאילו...
תעשה את זה בשיטה הרגילה פשוט.
אז הקבוע שלי...
כאילו אתה מעביר את המספר. לא, אבל אתה עושה את זה בשיטה הרגילה, פשוט תשיג את I בצורה פולרית שלו, ואז תוציא שורש. יש לך 4 שורשים בעצם. יש לזה 4 שורשים, לכל כאילו יש לך בעצם כאילו Z בחזקת 4 שווה מספר מרוכב, במקרה הזה המספר מרוכב הוא I, כאילו A0 ו-B1. נכון? אתה מציג אותו בצורה הזאת, אז אתה אומר כאילו זה האמפליטודה שלו, כאילו המקדם, ה-R, זה 1 בעצם, ו-EI